Hello, my name is Falco Bussi. I'm president of the Bruker Biospin Group. Bruker Biospin develops solutions for life science, for preclinical and clinical research, as well as for pharma, multiomics, including clinical phenomics and foodomics, and more. We are the clear market and technology leader for magnetic resonance analytical instrumentation, and we are practically present in every key life science research institution around the globe. Our focus today is the Bruker GHz NMR flagship technology in structural functional biology, with a deeper look in significant applications like neurodegenerative diseases, cancer, and COVID-19. What do these applications have in common? A key principle in biology is that structure determines function. In other words, the way a molecule is arranged, its so-called conformation, determines how it interacts with this environment and therefore how it functions. However, for these proteins to function, it usually requires a change in conformation. This is especially true for intrinsically disordered proteins, IDPs, which are a large and functionally highly relevant class of proteins, and they are involved in a large number of disease pathways. With an aging population in many regions on this planet, we also see an increase in age-related diseases, especially neurodegenerative diseases like Alzheimer's, Parkinson, Lewy body, and others.
What we now started to look at are actually these insoluble deposits of a protein called tau, which is believed to be the major culprit in Alzheimer's disease. So what we really try to understand is what is the structure of these tau aggregates, amyloid fibrils, and again, how does the structure differ between different diseases in the same way, because this will help us hopefully to target the right drug to the right person.
Our GHz NMR technology is ideally suited to investigate molecular interactions at an atomic level to obtain structural, functional, and binding information at near physiological conditions. This extends the frontiers of accessible research targets for pharmaceutical interventions, and by that, potentially novel therapies.
We just started now 3 months ago, but particularly impressed we've been by the quality and the resolution of the solid-state NMR spectra of amyloid fibrils of the Alzheimer-related protein tau, which we have started to measure and which really, gave fantastic resolution. So we hope to build on this in the future.
How does the Bruker GHz NMR technology enable and support research in neurodegenerative diseases? First of all, it helps in understanding disease mechanisms and pathways in a much more detailed level. Especially if you look at intrinsically disordered proteins like beta amyloid, but also alpha-synuclein. These are extremely relevant proteins in the disease process.
So this is in principle structure and dynamics of molecules. And we apply that to all kind of sorts of molecules in order to relate then structure with function eventually. Function is most of the time chemical function, but also biological function or pharmaceutical function. So if we understand that, we can understand how molecules interact with each other and modify their function. And sometimes, and this is one of the research lines, this is important for diseases. We are looking at how molecules behave under disease conditions and try from that also then to work on therapies for these diseases.
These proteins tend to aggregate during the disease process. Beta amyloid to form fibrils and plaques, and alpha-synuclein aggregates on the surface of neuronal cells and impact their function in Parkinson's disease. So to understand the mechanisms of this aggregation and identify molecules that can either inhibit the aggregation or even reverse aggregation after it happened are primary targets for novel therapies for these diseases. Future developments that will continue to support the research in the field and drive the effectiveness of the translation are, for example, software tools that significantly accelerate the analysis of these huge data sets which are generated by the GHz-class NMR machines, using artificial intelligence and deep learning to speed up the whole process.
It takes several years to get an outcome. That's scientific work. And it's like that. If you study such a system, it may take a half a year or 2-3 years. And what we recently started is to automatize this process. And we make a lot of progress. And it looks like that we need now maybe 2 weeks of measurements and 5 hours of computation, and then we are done. Now, this is possible for systems which are not as big, not so biologically relevant yet, but with the high field, we will be able to get there, and with the help of this high resolution, because you have a sharper picture, artificial intelligence is much easier to be applied, and so we will be soon automatize. That's my prediction, and of course it's my baby of research, and so I'm very excited about it, that we will soon deliver this, and everybody can get once a week a structure when a biologist works on a system, and not waiting a year to understand the structure and then try to understand what it tells you in terms of biology. And that will accelerate amazingly the process of rational approach in understanding biology.
Furthermore, creating a seamless workflow from the GHz-class NMR machines onto more standard systems, enabling the flow down of these research results into the drug development process.
Science is driven by developments in instrumentation. So we now have the best NMR spectrometer for biomolecular applications in the world. So it definitely will provide a competitive advantage.
Outcome could well be therapeutics and diagnostics for neurodegenerative diseases like Parkinson's and Alzheimer's. And if that was the case, it would of course be super rewarding, because it goes then from basic science to actually benefit to society.